Hello everybody. In this video I will demonstrate how we use polymorphisms to come up with haplotypes in lines and compare haplotypes between lines in different regions of the genome. We'll use this visualization tool, which is based on the IGV from the Broad Institute, the maize B73 genome as reference, and an iodent line that has a lot of coverage and which we will use as an example. Uh, the This track here is showing for the whole genome, so all 10 chromosomes, the alignment of the iodine reads versus the genome. And this part here is showing the results of the de novo assembly, so the scaffolds aligning to the genome. The track down here shows the annotation. Now we'll start zooming in into a specific region of chromosome 1, and we can start seeing the reads and the scaffolds aligning to the B73 genome. And if we zoom in more, we'll see the details of the alignments between the reads and the genome. I wanted to take you to a specific region that can demonstrate my point. If we go to this region, for example, and we concentrate on a specific gene, we can start seeing the many possible polymorphisms that are abundant in the alignment of the reads to the genome. However, as one can see, not all the polymorphisms are real. So in this case, for example, you'd see a lot of possible polymorphisms, so differences in the color between the reads and the genome, meaning differences in the read sequence or small insertions or deletions that do not really appear in the final decision of what is the sequence of this iodine line. Okay, so one needs to be sure that it filters out the right information before it decides on the right polymorphism that is the base for the haplotype decision. So in this case, a lot of those false possible polymorphisms will filter out and only a few were left. And now that we know that which are the true polymorphisms that which are not, we can base our haplotype decision on it. So now I will move to a polymorphism tab, showing a concentration of the polymorphism decisions on several lines. We'll zoom out so we'll be able to see different types of polymorphisms appear. And now you can see the concentration of the different types of polymorphisms for a specific line. This is Lancaster line 3, and this is all the tracks with the information about its specific polymorphism. And those lines here are showing the same type of information for an iodine line. So what we would like to do is see different types of polymorphisms, like SNPs, SNPs that change amino acid, SNPs that cause a stop mutation, SNPs, deletions, duplications, insertions, and so on. So now the task is to compare the pattern of polymorphisms coming from a specific line, in this case the Lancaster, versus the pattern of polymorphisms coming from another line, in this case the iodine. And the algorithm needs to decide, is this pattern of polymorphism identical enough to call it the same haplotype? Now I will move to allele separation tab, which codes the haplotype information in kind of a condensed and color-coded, so you'll be able to compare the sets of haplotypes between lines. The color coding is set in a way that lines that have the same color in a specific region will have the same haplotype, and different colors mean different haplotypes, and you can see how diverse and how variable is the maize information. So in that just a few lines here, you can see 1, 2, 3, 4, 5 haplotypes in the same area, and this is an indication of how difficult it is to work with the maize information without such a tool that really can give you a full view of what's happening in your genetic material. I'll now move to another set of haplotypes just to let you have the impression of what's happening in real the genetic diversity of the whole maize population. So what we see here is a high definition map of the local haplotypes in maize. Again, it's the same algorithm comparing the polymorphisms coming from the different maize lines. However, in this case, we used different types of information sources to come up with the polymorphisms. Some of them are NGS deep coverage or average coverage of the maize genome, and some are GBS data. And on the extreme, this tropical lines information here is coming from Ed Buckler's very low coverage GBS data that we were also able to use and integrate into our system. And as you can see, the complexity is really major. And on average, there were like 35 different haplotypes in those 76 lines, of course, representing very different heterotic group in maize. Interesting issue. If I move to just a subpart of the population, so it'll be easier to see. So a very interesting example in this case is compare this iodent line to this pH 3737 derived line, which is really a non-BSSS, and find that this area here is totally identical, meaning there are like 40% similarity in the genome between those very far lines from different heterotic group, and there's no known common ancestor. And so this is a really nice example of how you can use this tool to really find out what are the real genetic differences between your lines and base your intelligent breeding decision on knowledge about the distribution of the haplotypes between the different lines in your genetic material repository.